Good morning, everyone. In chat, I posted two links, one to each of the two slide decks that we're going to use today, and then just a reminder that we're going to start out with wrapping up what we didn't finish with the last lecture, which is talking about this command called unique, which you might intuitively have a sense of what it does. So you're going to need to log into Malibu to do that. If you're working from home, a reminder to VPN in. If you're on the university network, you're good to go. Let's see. I see. So reposted. Oh, that's terrible. The links are not available if you log in afterwards. All right. Let's see. There you go. All right. Okay, just a reminder, it's hard for me to see the Zoom interface and my slides and the console at the same time. So if you have a question and I don't see it in chat, which is seemingly common once I get going, please turn off mute and jump in. No big deal. All right, so we're going to start with this first lecture, which was what we covered last Thursday, pattern searching in genomes. And where we left off last time was my introduction to this powerful combination of commands called sort and unique. And the whole goal of unique is to be able to take an input file or an input stream and identify the unique elements in it. So as an example, let's take this toy file that we have on, you now have this file in your home directory on Malibu, so you should be able to.
You're not sharing your screen.
Oh yeah, thank you. That helps. Appreciate it. Okay. All right, great. So thanks for that. In your home directory, you should see a file called dna.txt, and if we look at the contents of it with cat, you'll see that this is a really sad little DNA file with a handful of lines of each of the 4 nucleotides. Okay, what if our goal was to count identify the unique elements in the file? So in this case, it's a bit trivial because we know there's only 4 distinct or unique nucleotides, but often we have no idea how many unique elements are in it. So maybe we have a file which is a line for each time a gene is identified as being associated with some phenotype that we're studying. And maybe it's thousands and thousands of lines long. Some genes occur 1 time, some genes occur 10 times, and you want on it what's the most frequent gene in that list. So the way that we do that is with the unique command. So unique, we could just say unique.txt, dna.txt, but unfortunately that doesn't really do anything different. It just reports the same thing. The hidden little secret about unique is that it only works if the data are sorted. So you have to sort the data such that all the occurrences of all the A's in this case come in a row, come in order, then all the C's, then the G's, and then the T's. So if we sort the DNA file, what you can see is all the A's, C's, G's, and T's come together. And then essentially what unique is doing, it says, okay, I'm just going to assume you've given me sorted data. If you haven't, well, that's your fault. You didn't know better. If you've given me sorted data, though, I'm gonna look through the lines, and every time there's a change between the previous element, the previous line, and the current line that I see, it will say, okay, I'm seeing a new pattern, and now it'll identify that as a new element. So, for instance, if we sort dna.txt and pipe it into unique, what we get is 4 distinct nucleotides because it identified this change right here, then it identified this change right here and reported C, then it identified this change and then reported G, And then when it gets to the last line, it realizes that that T nucleotide was the last element in the file. So now what I mentioned though is, what if we wanted to count the number of distinct elements? So not surprisingly, based upon the patterns we've seen so far, there's an argument called -C, which will count. So we'll report the number of occurrences of each of those nucleotides. Okay. This is trivial for this example because the file is really small. We can count this by eye. But the point here is that you can do this with a file that is infinitely, well, not infinitely long, but millions or even billions of lines long. The data just needs to be sorted. Once it's sorted, you can do this counting process very, very quickly. So it's very useful essentially for profiling or exploring what are the most common, least common entries in a file, and also for sanity checking. Sometimes you might want to, your expectation might be that every element in a file occurs exactly once, no more, or every element in a file, every entry or distinct thing like gene IDs occur more than once per file. So you can use the unique command to double check that expectation. Is that right? It's a control. Okay. So this is basically the example. All I wanted to say is that, you know, the unique, this combination of sort and unique is super powerful. You can use it. I use it daily in my work. Whenever I get to work on the command line, I know people in my lab and computational labs all over the world use this trick, this combination all the time. Okay. Any questions about unique? Okay, great. So at this point, we can shut down Malibu. I'm just going to disconnect. We're not going to be on there anymore. We're done. And now we're going to move from a Unix prompt into an R prompt. What I mentioned at the end of last class is that I wanted you to install RStudio. Hopefully every one of you has done that. If you haven't at this point, just follow along. And I'm basically just going to give you a crash course into how and why to use R. And I'll post the YouTube lecture probably tonight or early tomorrow, so you can go back and revisit it once you've installed R if you haven't already. Hopefully you already have. All right. So I've installed R. I'm going to have to make my screen, compromise my screen space a little bit here. Okay, and I'm going to get into the details of RStudio in just a second, but on my laptop, I just launched in applications this thing called RStudio. There's R, and then there's RStudio. What I'm going to jump into is why to use RStudio in just a minute. Okay, but let's revisit the basic motivation of the course and in part for using R. As I mentioned on day one, traditionally biology, especially genetics, has been considered a data poor discipline where experiments take a long time, they're very costly, and we're sort of limited in the data that we could collect. But because of next generation sequencing and other assays, improvements in microscopy and all sorts of things in biology, we are now often plagued with a completely different problem, in that it is easy to generate data, but our challenge now is often to interpret and analyze those data sets because they are complex, heterogeneous, and this is the most important point and will be a point of emphasis for coming lectures, where the data set has both biological signal, true biology in the experiment, in the results, but it's swimming in this vast ocean of technical noise. And what we need is abilities, data visualization, statistical methods, and basically just data processing approaches to separate out the true biological signal from the technical noise, such that we don't publish articles that are basically irreproducible because the thing that we thought was signal was actually noise. Okay. And I think Albert Einstein's quote really summarizes this conundrum. So with that goal in mind, this is where we're entering the domain of data science and visualization and statistics. And I think one of the things that I try to emphasize to myself, uh, to people that train in my lab, and to anyone I can tell, especially you all, is I think our responsibility as scientists, and this is ever more true with computational work, especially in genomics, is to really know our data. And to know thy data, I think you begin to inherently distrust your results. You should. Don't get fooled into inherently believing that your data support your hypothesis simply because it looks like it might. It looks like it might. It could look that way simply because there are problems in the data that happen to manifest in a way that support your hypothesis. So one of the first things that we must do when looking at our results, when scrutinizing our results, I should say, is to look at it, to visualize it. And that's everything from basic scatter plots that are shown here to more sophisticated visualizations for quality control and things like that are germane to RNA-seq and ChIP-seq and mutation detection and all sorts of things that we do. And a very famous example of this is something called Enscom's Quartet. So what's shown on the left is a plot of the quartet. And each of these 4 plots is a scatter plot of different XY coordinate pairs. The source of the data doesn't matter. It was completely simulated to demonstrate a point. What you can see is that visually, the data are very different. The distributions are incredibly different. This one has this very vertical pattern where Y varies, but X doesn't, except for this one point. This one looks largely linear, but with one outlier. This is linear, but with variance about the slope. And this is, some superlinear pattern. The point that Anscombe tries to make with this is that if you didn't look at the data, you would be fooled into thinking that these data sets are essentially identical, because each of these data sets, perhaps surprisingly, have identical mean, variance, and correlation. So if you're comparing experiment 1 to experiment 2 and you don't look at the data and just measure summary statistics like mean, variance, and correlation, you would be fooled into saying, right in your paper, that these were reproducible, very similar data sets. But if you plot them, you can see quite clearly that they're not. So the Anscombe's Quartet is used in data science and data visualization as sort of a baseline example of why you must look at your data and do not trust summary statistics. Look at the actual distribution of the data. And just as an aside, this is why journals are ever more requesting specific types of plots instead of more traditional plots. So an example is, instead of box plots, journals are often requesting that we plot things called like swarm plots. And there's another one that I can't remember, violin plots, swarm plots, because things like box plots actually hide multimodal distributions. And we'll talk more about that later. Box plots kind of assume that your data are normally distributed, but if there's 2 different modes, the box plot doesn't show that well. In contrast, plotting a histogram or plotting a swarm plot or a violin plot shows that multimodality in a much better way. So it's closer to the actual data rather than these more abstract plots that hide subtleties in the distribution of the data. Okay, so why R? There's lots of different languages where you can visualize data. One of the languages I use most is Python. You can make plots in Python. I recommend using R simply because for a couple of reasons. One, it is a more comprehensive statistical analysis language. It's really good for statistical analysis. On the other hand, it's not a great general purpose programming language like Python, but it is very good for stats and for data visualization. More importantly, why R and not something like Excel? Excel, in many ways, tabs in an Excel file, those worksheets, are very similar to data frames, which you'll learn about on Thursday when you go through the slides on Thursday, which are basic data frames, are the same conceptually as spreadsheets. There's rows and there's columns and there's data values in them. Okay, so why R and not Excel? Well, let me go through the reasons. First, it's free, and I guess that argument is a bit moot because Excel is largely free now, and you could use something like Google Sheets. You don't have to like pirate it or anything anymore. If you work at a university, you can usually get it for free. I think this is an important one. R is an open source piece of software. It's used by people all over the world. It's free, and there's lots of very talented developers that are creating new statistical packages and data visualization packages all the time, and they're doing it for free. And there's an enormous user base. That means that once you get your hands dirty a little bit with R and know the basics, but you're out on the bleeding edge of what you don't know, you can go to things like Stack Overflow or Stats Exchange or Google to figure out, if you're facing a problem, chances are someone else has already faced this problem and posted a nice example. So I've mentioned this before, but a dirty secret about programming and stats and anything is spend a lot of your time just looking for prior art. I think these last 2 points I would say are the most important though. The difference between R and something like Excel is that you're writing code that can be executed once it's right forever. So you can reproduce an analysis from top to bottom by saving an R script, which we'll get into over the course, that once you leave your lab and go on to a postdoc or postdoc to a faculty job or a biotech job, that the legacy of what you did lives on in that script. So a new grad student or a new analyst can come in, take that script and reproduce the same thing. That is not impossible in Excel, but it's excruciatingly difficult. Excel is really good at just, it stores a spreadsheet and there's plots maybe in your spreadsheet, but you had to do a bunch of clickety clicks to make the plot, and that's not reproducible unless you like literally write down the script of someone right click on this and then click on that tab and change the awful default colors in Excel to this and then do this and this and this. So it's not reproducible. The last thing I want to emphasize is that R does a much better job of handling large data sets that we tend to deal with in biology. Now, this is not to, completely bash Excel. It has its place. It has its place for storing, being a manifest of an experimental design or something like a system of record, but it's not a good solution for analysis. So I would encourage you, one of the goals of mine in this course is to get you comfortable enough in R, if you're not using it already, to make it one of the tools in your quiver for analysis, okay? Just a quick, so I have a sense of what we're getting into here, how new this is going to be to everyone. There's a feature, what is it, raise hand? Could you just raise your hand if you've ever used R before? All right, that's great. So it looks like about roughly half the class, maybe even more, has used this before. All right, well, for those of you that haven't, this is going to be new. For those of you that have, obviously this is going to be a little bit of review. Hopefully I teach you something new today, but I promise you over the course of the next few weeks, we'll learn some new things for all of you. But if if there are for those of you that haven't used R, use Slack if you have problems. And those of you that have used R, outside of the homework, please help answer questions. That's a great resource. Okay, so thank you for that. R has tons of packages. This is just a graphic from rgraphgallery.com, which shows all the different analyses that you can do in visualizations. These packages range from things to make cool plots to things to do a t-test or to do a generalized linear model or lasso regression or gosh, I can't even think of all the things that you would want to do, but basically any statistical method, especially in biology, is available in R. And for those of you that work on single cell genomics data, all the best analysis tools in actually bulk RNA-seq and single cell RNA-seq are essentially developed in R. The interfaces are there in R. Okay. One aside, this is bashing Excel. One of the reasons that people tend to use R, not Excel, is you may have faced this. One of the genes I worked on before I went to grad school, I did a year of post-baccalaureate work because I was trained in computer science and didn't know any biology. So I went and worked in this lab that studied OCT4, which it's also known as POW. I forget the numbers after POW, but the more common name for it is OCT4, and it's implicated in cancer, but it's also one of the genes that was in the lentiviral cocktail used for creating not embryonic stem cells, but converting differentiated cells into progenitor cells. Why did I say all that? Well, OCT4, if you put OCT4 into Excel and hit enter, depending upon how you have Excel set up, the first thing it will do is automatically change that to October 4th. It'll change it to a date. So it botches data files all the time. There's another one called a gene called CEP7 in human genome that gets converted to September 7th. And there's MAR something that gets converted to March. R doesn't do that stuff. So this paper talks about is how many published manuscripts have supplementary tables and figures that are saved in Excel, and the results are botched. You've probably seen this by now. Okay, so R programming language. This has been around for a while. It was conceived in 1992, and the initial version of it was released in 1995, and then sort of the formal, stable version of it was released in 2000. I was working at a bank in San Francisco in 1999, 2000, and when this first came out, there was already talk of people switching over from these incredibly expensive statistical packages like SAS and Stata into using R because it was free. It was created by Rasa Haka and Robert Gentleman. Some of you may know Robert Gentleman. He then went on to be a part of Genentech, and now he's on the board and works at 23andMe. He's an accomplished scientist and on his own, interested in cancer genetics, but also turns out to be a really good statistician and programmer. The name for R came because it's a play on a predecessor called S, which maybe S was for statistics, and why they didn't choose T instead of R, because T comes after S, I don't really know. But R is the name of the language, and now there's this now played out cliche of often naming packages for R, like tidier and limmer, and playing on the R at the end and capitalizing it. Okay, there are many ways to get R. You can download it. If you just Google download R, you can find it. That's not the recommended way. This is what I learned R on, was this sort of clumsy looking interface that is reminiscent of, the prompt is reminiscent of the Unix prompt that we've already used. The way that I would recommend is using RStudio. So RStudio is supported through licensing. So they license RStudio to companies and to universities. There is a free version, but what's really nice about RStudio is the licensing fees that they get from institutions help support the salaries of really good developers that make RStudio into just a fantastic piece of software for data analysis. Okay, so let's switch over. If you haven't launched RStudio already, please do so. I imagine most of you that have experience with R use RStudio, so this will be new. This will be nothing new. But what I'm going to do is I'm going to switch over for a second. I'm going to switch between these two windows. What this slide is highlighting is that RStudio by default is broken up into 4 main panels. There's the lower left one is the console. Let me do something here. Okay, so down here on the bottom, you can see my prompt. I'm hitting enter a bunch of times and I can get a new prompt. If I want to clear my history in Unix, you type clear, then enter. In RStudio, you type Control L, and that'll erase all the junk. You can also use the up arrows to scroll through previous commands. Here's some attempts at a plot that I'm making for a grant that continues to not be what I want, but that's my attempts at getting to work. So you can use the same tricks that we talked about in Unix to be lazy and go back in history. You can also do Control R, just like in Unix. RStudio has adopted some of the conventions from Unix, so there's parity, I guess, between the two. So if I do Control R, I can visually scroll through all the different commands. I can hit escape to get out of there. Okay. The top panel is where you work on a script. So you can see that this has a file saving interface. So I can type a bunch of commands here. These won't do anything. I can do a linear model of X and Y. That's backwards, but doesn't matter for now. And I can make plots, etc. Now the key point is ultimately I can save this, and I can call it, broken script.r. And once that thing works, it'll always work, with a couple of exceptions. Good conventions for an R script would be to document where the starting file was on your file system, or ideally have it set up so that the first part of the script is to navigate to the right directory where the file is and establish that as the working directory. We'll talk more about this later, and then read in the data and do all the analysis. So with caveats like that, making sure you set future users up with the exact same working environment that you had, the script is reproducible. Okay. But as we're trying to figure out what commands go into the script to get things right, which is where we spend most of our time, we're usually doing that on the prompt, okay, on the console. There's the environment tab, which is the upper right, and this tells us all of the different data sets or data frames or variables or whatever that we have loaded into our environment. I have saved this environment over the last couple of years, so there's some old data sets that every time I load RStudio, there's some old data sets that come up, and I can take a look at. So from some work from previous students in the lab and some ongoing work. What's really nice about RStudio versus R, let me find one that might be useful. If I click on this data frame in the right-hand side, in the upper left-hand, it shows me the contents of that data frame in row and column format. So this is a data frame of germline mutations in some families that we studied from a few years ago. But this is essentially a data frame, and this allows us to use our eyes to just have a visual scan of what the data set looks like. Another way to do that is you can use the view command. The view command will do the same thing. So you can click on it or use view. All right. Now, lastly, in the right-hand side, often what we're doing is making plots in R. So if I wanted to, let's say, plot F1, let's see if I can, age in F1, oops. So the age of the father against the number of de novo mutations that are observed. We can make a plot, and it shows us this plot in the bottom hand side. And what's nice is if we then make another plot where we make the color, my favorite R color is Dodger blue. I'm not a Dodgers fan, but I think it's a pretty color. Now what we have is this back arrow where we can scroll through our history of plots. Okay. All right, so that's the RStudio interface. The main distinction with traditional R is that in traditional R, you just get this console. So this is much nicer for interactive data visualization and processing and saving scripts and everything. It's a huge advance. All right, so just like the Unix prompt, this R prompt, which is denoted by this greater than sign, it's bored. It's just sitting there blinking. That's what this cursor is, just blinking, bored, waiting for us to do something, to give it some instructions. Okay. And you know what I'm going to go through now is some basic commands that we can do in R just to get our feet wet. So we can do basic math. One thing I should show you quickly, if you don't have RStudio loaded, if you have R, if you install RStudio, you have R on your computer as well. So from the Unix prompt, you can type R and you get a prompt. And if you want to do math, it'll take, for me, it takes a little bit to load up because it has to load up all these data sets that I have. But you can use that to just hop quickly into R to do math on the command line if you need to. Okay. All right, so we can do 1 plus 2, we can do 1 times 2, we can do 1 divided by 2. It does, it works the way you would expect. One of the things that is a little confusing is what this means, what this bracket 1 means. It basically means that there was one result, one line of results, one row of results. This is important because some commands or some packages will generate multiple lines and columns, so you can use these as ways to refer to specific aspects of the result. So you can refer to line 2 or line 1 of a result. R's got a lot of built-in functions. So by default, you know, often in RNA-seq or in biology in general, we tend to convert things to log scale to compress an enormous dynamic range of values into a more a narrower range of dynamic values for visualization. There's other reasons to do log tricks, but we can just say log 10 is a so-called built-in function. Okay, so 10 to the third is obviously 1,000, and we can just go on and so forth. I think the default, let me check myself, pretty sure the default log is natural log. Yep. I don't know if there's a built-in log 2. Yep, there's log 2 as well. So 2 to the 4th is 16. So that's great. We've got base log 10, base 10 log rather, log based on Euler's constant, and then log 2 as well, just built in. Right. So let's move on. So one of my favorite tools in R is to build just toy data sets to play with what functions do. So I often, when I'm learning something new or want to do sort of a basic simulation or prototype for a plot or a prototype for analysis, is to just generate some toy data. So there's this built-in function called C, which is, I think of it as for combine. I can, let me just steal this from the, I don't need to type, so let me just steal this. If I type that, it'll report a vector of things, a vector of integers in this case, 1, 2, 3, 1, 3, 5, 25. If I want to save that into something useful, I can assign that vector to some variable. I'll just actually call this v for vector. And I can say v equals this list. And now if I type v on the prompt, it'll report that list. But now it's saved. In contrast, if I just use combine, it'll show the results of that, but not save it anywhere. It's disappeared into the ether once it's spit out on the screen. All right. So I can now I can do interesting things like take the log 2 of v. And this is where I think one of the best features of R is apparent. R is a so-called vectorized language. It means that what that means is when you apply a function to a list of things or a vector of things, it knows inherently to apply that function to every element in the vector. This should be the same for a data frame, the same for a matrix, all sorts of things. So that it allows you to do very powerful analyses on a large number of things with very minimal code. If you come from a more traditional programming background like C or Python or things like that, it's actually a bit of it's a little hard conceptually to think about, because in those languages you would have to write loops to do this type of thing. Okay. So I already jumped ahead a little bit here and assigned this combination, this list of 7 integers to a variable called v. Let's be consistent here, and I'll call it my numbers now. There's 2 conventions for assigning things to a variable to save them for future use, for plotting or for statistical analysis. I think the traditional is this arrow. It's a less than sign followed by a dash. And the idea is we're taking this data and putting it in this. So the arrow is like moving it into this variable. It's very painful for me to use that. I tend to use an equal sign because that's, first of all, it's the same. And that's the convention that's used in basically every other programming language. So I just like to use an equal sign. If you want to use the greater than sign followed by a dash, go for it. It's your choice. They're equivalent. All right. So as I said, now that we've saved this to a variable, we have access to them in our script, and it's reproducible. We can play with this now. So the cool thing about R as well is we can access data frames or simple things like these lists or vectors by position. So what do I mean by that? Well, if we wanted to look at the 3rd element, figure out what the number is in position 3 of this vector, we just say my numbers of 3. So this open bracket, yeah, that's a bracket, right? Yeah, open bracket, and then a number followed by closed bracket says, hey, open up this my numbers vector and look at a particular element. And the element I want you to look at is position 3. So R is a bit different than other programming languages in that these positions in the vector are 1 based. So the first element is 1, the second element is 2. So it follows more of our I think human way of thinking, but other programming languages tend to use the first element is 0, the second element is 1, third element is 2. So there's 0 based indexing. R is 1 based. Okay, so third element is 3. Let's see what the first element is. Yeah, that checks out. The first element is 1. The 6th element better be 25. Oops. Oh, yeah, sorry. The 7th element is 25. Okay. So Oh, shoot. I see there's a bunch of hands raised. I'm guessing that's from my question 15 minutes ago and not ongoing questions. Yeah, okay, cool. All right, thanks. Right. Now what we can do is now we can start to do some analysis on these numbers. So we can compute the mean of these numbers by just simply running the mean function. And the cool thing again, this is that this vectorization in R. It knows to automatically essentially unpack all the different numbers that are in this vector, sum them up, and as I'm summing them up, counting how many numbers it's seen, and then dividing by that. Mean is, the sum divided by the number of entries, right? So we get this nice mean. We can, I think, compute the median. Does R have median? I think, yeah. The median number is 3. We can do max. It should be obvious. We can do min. Okay. We can plot my numbers. It's going to be kind of boring. So the x-axis is just the index number in that list. So the first number, the second number, the third, all the way to the 7th. And you can see that, this follows exactly what we saw in the numbers. The last one is really high, right? And we can also do things like we can plot the log 2 of those numbers by combining functions. So now the plot function is the outer function, and then we can apply a modification to each of those numbers. So again, vectorization, it is for every number in the myNumbers vector, it's taking the log 2 of it. So that generates behind the scenes a new vector, which is 7 numbers that are the log 2 of the original numbers. Those are handed off to the plot function and plotted. Okay. And what's nice is in R, it automatically changes the y-axis to be essentially the function that we used for the y value. Sorry. Any questions about that? All right, this is probably review for the R gurus in the course, and I'm not an R expert, so I will probably learn things from you along the way, but hopefully I teach you some new things. If we take, this is just a toy example to say mean. Well, if we just apply mean on its own, it's going to give us an error because it needs to calculate on something. We need to give it an entry, some values. So that's why we need to give it either my numbers, or we can do things like this, I think. Yep, we can just give it, no, that didn't work. Let's see. No, that didn't work. The reason is we need to make this a list with the concatenate or with the combine operation. So the combine operation makes those numbers 1, 2, 3 into an actual vector that the mean function can work on. There's some idiosyncrasies like this in R that take some getting used to, for sure. All right, so just like the man page in Unix, there's help functions for there's a help interface for every function. All you need to do is say help and then type in the function name, and it will automatically over here in the bottom right give you a list of topics that might be relevant to what your search term was. Here's a typical R help page. I find them to be a little confusing because it's this constant redirection to different depending upon what you search for, you end up going through this sort of rabbit hole of looking at help page after help page. But eventually, in this case, we get to a description that sort of talks about what the parameters are to the mean function. X is the vector that you use. Sometimes we have to deal with unknown values or NAs. We can either try to work with those NAs or not. And then often, I think this is the most helpful thing, there's typically an example, okay? So let's actually take this example and see what it does, because I find it pretty useful. So in this case, we're creating a new variable. I'm actually going to keep our same, let's create a new one. My numbers 2, I'm going to ditch it, make an equal sign. This is a fancy use of the combine function. It combines two things. This is called a range from 0 to 10. So that basically automatically generates that shorthand for saying produce every integer between 0 and 10 plus the number 50. So we can check, now we can look at what the contents of that actually is. And so we get the numbers 0 through 10, as I said, plus 50. Now we can take the mean of that, as is shown in the example. So this introduces a new concept. If you wanted to do a simulation or an analysis of values, let's make a more sophisticated plot. I want to see what a scatter plot looks like, where I take the numbers 1 through 1,000. So let's say x equals 1 through 1,000. And I want to then compare what the relationship with the log 10 of those. So I want to do the log 10 of x. So here's the beauty. In just a few keystrokes, I've told the computer to generate the numbers 1 through 1,000. I didn't have to sit here and type C, open parentheses, 1, 2, 3, 4, etc. We can use these shortcuts to do that for us. And then, because we get vectorization in R, it will take those 1,000 numbers and automatically compute the base 10 log of every one of those values and store it as a list in Y. So how many values are in this vector Y? Anyone have a guess? No guesses? Okay, well, the length, there's 1,000 entries in X, 1 through 1,000. If we take the log 10 of all 1,000 of those, we get 1,000 entries in Y. Okay, so let's look at this. Now we can use the head command, just like in Unix. We can look at the first 10. Well, I guess it's not the first 10. I always thought it was the first 10 entries in X. And we can look at the first 10 entries or the first 6 entries in Y. So the base 10 log of 1, yes, is 0. The base 10 log of 2, I can't do that in my head, but apparently is 0.3, and so on and so forth. So now what we can do is just do a scatter plot of X against Y. Now we see this expected logarithmic relationship between X and the log of X. Okay? The whole point of this is you can simulate or do some prototype analyses or prototype plots really, really quickly by using these shortcuts to just generate toy data. Okay? All right. Oh, we covered that. Right. I find this function summary to be incredibly useful. It creates summary statistics for the vector that you give it. So it tells us the minimum, the max, the mean, the median in the 25th percentile and the 75th percentile. You would have to run 5 or 6 different commands to get each of those. Not only that, you'd have to remember what those commands are. The beauty of summary is you just have to remember one function name and it gives you all this information in one go. So now we can do this for so it did that very quickly for 6 numbers, or 7 numbers, I guess it is. But what about for 1,000? It's nearly instant. We know that my number started at 0, and oops, let's do this for x actually. X has 1,000 numbers in it. So now we know the minimum is 1 because we started at 1 and went to 1,000. The max better be 1,000. It is. The median, not surprisingly, is close to 500, but it's not 500 because we have an even number of numbers. So it has to be the middle between what 500 and 501. And it also reports the first quartile and third quartile. Super nice function for basically, this is what I would call exploratory data analysis. Like, what are the properties of my data? I've loaded up some file from an experiment. You know, we're working with toy vectors right now, but the same process applies to data sets that you load into R from your own experiments. You want to make plots of it, basic summary statistics of the data therein. Okay, so another function that I find really useful is table. So table, I'm really glad that we covered unique and unique.c at the beginning of the class, because table is the R analog to unique-c. So it will go through, oops, I almost typed unique. It'll tell us each of the distinct numbers is the first row. So we remember that there was 1, 2, 3, 5, and 25. Now let's compare this. So there's 1, 2, 3, 1, 3, 5, 25. Now look, then the second row is how many occurrences there were. So there were 2 occurrences of 1. Yeah, that checks out. 1 occurrence of 2, 2 occurrences of 3, 1 occurrence of 5, and 1 occurrence of 25. Okay. So this works out really nicely. And again, looking at the frequency using the table command to look at the frequency of values is a really useful approach for quality control and just sanity checking. You know, what if you saw a gene in there that every other gene occurred like 10 times, but then there's one gene that occurred 1,000 times in some analysis. Is that expected? Is that weird? You would never know it unless you looked. So this is, again, exploratory data analysis for just sanity checking. Okay. So how would we create another vector of numbers? Well, again, the function that we use is combine. And we can take, let's create this new vector, your numbers. So I have my numbers, you have your numbers. So now let's double check it. Oh, another thing I should mention is there's autocomplete in RStudio, just like on Unix. So if I type YO and hit tab, it'll automatically complete to a variable name that is distinct up to that point. So if I do MY, it's going to give me in tab, it's going to give me 2 options because there's 2 variables that start with MY. And then I can use my arrow keys to pick which one I want. Okay, so there's really nice, that's another nice shortcut if you're a bad typer like me. All right, so again, now we can plot. I'm going to give you a little bit of a crash course into plotting at this point. Many of you, I imagine, use ggplot. We're going to talk a little bit, we might talk a little bit about ggplot later in the course. It's a really nice package for creating fairly pretty plots. I'd say the learning curve is a little bit steep. And one of the problems I have with ggplot, although I use it all the time, is that it's really great for basic plots. It becomes quite difficult to manipulate plots to do very specialized things, say for publication. So I'd like to introduce the basic, what's called base R plotting functionality before we get into the more sophisticated stuff from ggplot and the like. All right, so now the basic use, a basic use of the plot command is a scatter plot. So what comes first is your X values and then your Y values, okay? So let's just try that. Right, so we've got a scatter plot of numbers, and these are now each value on the x-axis is the first value from my numbers, the second value and third value, and then it matches that up with the first value in your numbers to place this point on the scatter plot. Okay. As I showed, we can use the column or the color, COL, to change the color. So another nice one that I enjoy is dark red. We can change the size of these points. Oh boy. I think that's like maybe 5. Nope. Oh, what is it? Anyone remember? Let's see. Oh yeah, CEX is size. So those are horrifically ugly large points. There's another one that is the shape, and I never remember this. If anyone remembers the shape, let me know. But I don't, so I'll ignore it. The other thing we can do is we can add specialized labels. So we can say X labels is random numbers set 1, and then we can say the Y label is equal to random numbers set 2. And that changes the labels. Okay, nothing too fancy. All right, so moving ahead, what I want to show you now is a couple of other commands that we can do with R, and because we have a little bit of time left here. So one thing that we often want to do is not only plot like a scatter plot like this, but there's other types of plots that we can do. One of the more common plots is a histogram. And just like the plot command, there's a function called histogram. But to make a histogram, typically when you're making a histogram, you have more than just a handful of numbers like we have in these silly 2 vectors, my numbers and your numbers. So what I can do, we can generate some random data. One of the things that we can do with R that's super powerful is we can generate random data. So there's a bunch of functions that start with R, and those functions generate random data. And then the other aspect of each of these functions is the underlying statistical distribution from which the random data are generated. So for instance, we can generate random data from a normal distribution. And so the function to do that is rnorm. And if you're familiar with the normal distribution, we'll talk about this more. It's also called the Gaussian distribution after Frederick Gauss. The normal distribution has 2 properties. It has a mean and a standard deviation. So you notice here, let me do the help on rnorm. If we look up the help, yeah, so this is one of the confusing things about when you look up rnorm. It actually gives you help on 4 different functions, one of which is rnorm. This is the one we're looking for. It expects 3 parameters. Let's talk about the first, the second, the last 2 first. We have to give, when we're generating random data from a normal distribution, it needs to know the mean of that distribution and the standard deviation, because those are the 2 parameters of the normal distribution. So you probably know that in a normal distribution, within 1 standard deviation, something like 67% of the data is within 1 standard deviation of the mean. I think it's 95% within 2 standard deviations. And I'm challenging myself here, but I think 99.7% within 3 standard deviations of the mean. So the number of standard deviation, the size of the standard deviation governs the variance or the width of that distribution. We'll see that in a second. So, and okay, and then the first parameter is how many random numbers do you want to generate from that distribution? So let's start out with something kind of simple. Let's start out with 10 randomly generated numbers from a normal distribution. I'm going to name the parameters here so it's a little clearer what's going on, with a mean of 1 and a standard deviation of 1. So these are the 10 random numbers that it generated. And you can see that they're sampled. They seem to sort of be on both sides of the mean is 1. So we should expect some values less than 1, like this one, and this one, and this one, for example, and values greater than 1. Now, the beauty of this is it's so fast. I can generate 10,000 numbers in just the same amount of time. And it doesn't even attempt to show all of them because there's just so many numbers. But what we can do is let's create a new variable called, let's call them normal distribution numbers. Okay, so we can now create a histogram from those norm numbers. And that histogram should look something like something approximating a normal distribution, because if you're familiar with the histogram, what it's doing is counting up, basically looking at the frequency of each of values that are in a particular range. So it's kind of like the table command behind the scenes, where it's looking at all the different occurrences. But because the Gaussian or normal distribution is a continuous distribution, there's an infinite number of possible values. It has to break up the values into ranges. So like how many numbers were there between 0.5 and 0.6? And that will be the position on the x-axis in the histogram, and the count will be the number of values that were observed in that range. Okay, so here's what we get. We get something that is pretty symmetric looking. You can see that the ranges are pretty wide. There were 10,000 values, but it chose just eyeballing this, it looked like, so there's 5 bins between 0 and, there's 4 bins between 0 and 2. So that tells me that the width of each of these bins is roughly 0.5. Okay, we can play with that. I think it's breaks. Yeah, breaks, we can make it 20, and this will be a finer, oops, that's the default. We can make a finer scaled histogram by just, if there's 40 breaks, it chooses 40 evenly sized ranges from which to count the number of values that are observed in that range. So if 20 breaks gave a range of 0.5, 40 breaks should be something like what, 0.25. Okay. We can also, with the histogram, we can change the color. Go back to, let's try green. I don't know what that'll look like. That's pretty ugly. Let's go with dark green. I just can't stand to look at that. All right, that's better. And again, the functionality is the same. We can, I think it's title. I think we can call this hist of random normally distributed. Oh boy, this is embarrassing. You guys get to see how terrible I am. Numbers. Okay, no, what is it? Not title, main. I always forget. This is where I end up having to Google. Main. Oh, blew it. All right, hold on. If I fail this again, no, I didn't. I got it the second time. Another way would be to just, again, look at help hist, but I'm stubborn. But Yeah, somewhere in here, there's the main. It'll tell us the title. Okay. Right. So what else can we do? Another tradition. So this histogram and scatter plots are two of the sort of workhorses of basic data visualization, where we get a new data set loaded into R and we want to inspect our data. So if we assume that our data are normally distributed, the first thing we should do is take, if you've got a data set of mouse weights in two different conditions, or time required to observe some behavioral phenomenon over lots and lots of observations, you might have some intuition about what the distribution of that data should be. Well, check that. Load it up into R and plot it. Make a histogram, make a scatter plot, whatever's relevant to the data type, and check that your intuition is right. And I can't count the number of times where just a simple sanity check like that have revealed something very fundamentally wrong about our assumptions, A, or very fundamentally wrong about the experiment, such that the observed values are nowhere near the expected values. So this last slide that I didn't touch upon before is that, R is really powerful, but again, it's stupid. Like if we plot this, if we type this, it's like waiting for us, and it tells us it's waiting for us to complete a command by the fact that there's this plus sign. So anytime you see a plus sign, that means that the command that you thought was right is incomplete. And it's pretty clear that I'm missing this closed parenthesis. So as soon as I complete that, it generates the plot and gives me my prompt back. All right. Well, we have 15 minutes left in class, and I would like to move on to one other topic in plotting, but I wanted to just make sure that there's any questions at this point. Is everyone roughly with me, and anyone having problems? Are we all on the same page?
Same page.
Okay, great. Awesome. Okay, so the last thing that I want to show you is basically how to do more sophisticated plots than histogram. And I mentioned that histograms or data are often multimodal. So I wanted to generate some multimodal data first. So how would we go about doing that? What we can do, the way I would do this, it's kind of a hack, but I'll show it to you. So we're going to generate random numbers. We're going to create 1,000 random numbers with a mean of 1 and a standard deviation of 1. And I'm going to call this A. That's our random number set A. And I'm going to create another 1,000 numbers with mean 10, same standard deviation. I'm going to call that B. And I'm going to create a new data set, which is called my, multimodal. And I'm going to make a combination. I'm going to combine A and B. So at this point, multimodal has 2,000 values in it. 1,000 generated from this random generation function, and another 1,000 from this function. But the properties of these numbers are fundamentally different. This one has a mean of 1. This has a mean of 10. So the numbers on average in B should be roughly 10 times the numbers in A. But now, so let's imagine that in our experimental condition, like A is cases, B is controls. But we basically collect all that information. Or actually, let me make a better analogy. Let's say that A is the result of experiments done on Fridays, and B is the result of the same experiment done on Mondays. So I would say Monday, at least for me, I do better work on Mondays than I do on Fridays because I've had time to recharge over the weekend. And maybe the intuition is that the experimental results from B are more in line with reality than on Friday when I'm doing this experiment at 4 o'clock and excited to maybe go home. Let's imagine this is non-COVID days when we're in the lab and we get to go home and relax. So maybe we rush the experiment. But the data, we aren't really tracking. We aren't tracking, maybe our lab notebook isn't great and we haven't tracked the date. And so we ultimately have this data set and a spreadsheet or a text file, which is the result of 2,000 experiments. You've been working hard, and they're all lumped together. So we're not able to distinguish A and B, but behind the scenes, the truth is that there's really these two batches of experiments. Batch A, Mondays, batch B, Fridays. So what I'm trying to illustrate here is that there's actually a so-called batch effect in our experimental data. So I'm simulating that now. So if let's look at the histogram of this multimodal data set. Now I can see it, right? Let's look at the mean of this multimodal data set. Yeah, that's definitely the mean. It's right here. But that mean, just that summary statistic, belies the complexity and the batch effects that are in the data. So this is getting back at Enscom's Quartet. Summary statistics alone fool you. So let's maybe look at the standard deviation of this multimodal data set. It's large, but in my mind, if I just saw mean and standard deviation, those two numbers, I would think of something like this. Let's create a third data set called C, which has a mean of 5.5 and a standard deviation of 4.6. And I do a hist of C. Oops, if I can type it right. Okay, that's if someone told me, oh, our data have a mean of 5.5 and standard deviation of 4.6, that's exactly the picture that would come up in my head. 5.5 is the mean, and then there's this big standard deviation. Whereas in truth, the data look like that, right? Okay, so histograms are not often used in papers. Well, they are being used more frequently now because people are sort of onto this problem, and journal editors are onto this problem and enforcing other types of data, other plots like histograms. But box plots are more traditionally used, especially when comparing two conditions like mouse on a normal chow diet and mice with a differential diet, because it fits really nicely. It's a simple plot. It's small, it's digestible, it's easy to fit in a manuscript, right? The problem is that a box plot of multimodal data like this really hide what's going on, whereas a histogram will actually show it. I forget, honestly, maybe this is where I could solicit help from others. I can't remember if there's a built-in box plot. I think there is. Looks like there is. So now we can see that, I hope. Yeah, so there's a box plot of that multimodal data. Does that look multimodal to you? No. It looks like it's just very noisy experimental data with a lot of variance about some median value. So there's other plot types. One is called a swarm plot, and we're going to revisit this later, but I kind of want to close out with this thought. Let me show you a swarm plot. Let's find it for R. I'm just cheating and using R here. Okay, I think I've used this one before. I don't know what a waffle plot is. So let's just go with this. All right. Yeah, here we go. So, boy, maybe I could, maybe in the last little bit, I could try and, let me see if I have this library. I do somehow, miraculously. So I think maybe I can do B swarm. If you, we'll talk about how to install packages later. If you don't have it, probably don't. Right? So isn't that's so much better? It's almost as concise as a box plot, but man, does that really show what the data truly look like. So if you were, box plot where the medians might have been shifted, a t-test, which we'll talk more about later, might have said that the two conditions were different. But if we knew the properties of the data, like I think as reviewers or people doing journal club on a paper like that, we'd be a little skeptical. Okay, any questions about any questions about B swarm plots, histograms, or anything I covered? Hopefully that gives you a sense of how you can be fooled by your data if you don't look at it very, very closely. All right, so just to give you a reminder of Thursday, unfortunately, I'm going to be on a conference call all day for grant review, so I can't be there Thursday. But I've posted on the lab website, or sorry, the course website, on Thursday, you want to go through this slide deck. And just to give you a sneak peek, this is building upon what we talked about today. This is borrowed from another interest group that I ran for a while. It's talking about data frames. So data frames are essentially the equivalent to spreadsheets. And I would say that data frames are the more natural way of taking data from an experiment and putting it into R. So a common workflow, just to set the stage for what and why you're going to go through this on Thursday, a common workflow is to use commands and tools on the Unix command line to process data and get it down into a tab delimited file, text file, that's, that represents sort of the final data set that you want to do analysis on. That tab delimited, those are columns. Rows are usually observations or experiments. That tab delimited text file is the conceptual equivalent of a data frame. A data frame in R is rows and columns. So common workflow is to get it into that final text file and then start in R by loading up that data frame into something we can work with in R, where we can then start making plots of all the observations on rows and plotting the values that are observed in the columns. So things like experimental conditions or males versus females or case versus control or experimental versus wild type, whatever. So this will take you through some basic concepts of what data frames are and how to use them. And that'll be important because we'll be building upon those concepts when we start talking about RNA-seq. Also, there are some incredibly good packages now in R for working with data frames. So I would encourage you to think about trying to get your data in a data frame format once you go through this for analysis in R in the future. Okay, so we've got a minute left. So I'm happy to wrap up, but if there's any burning questions, let me know. Okay, great. Well, I apologize I can't be there on Thursday, but hopefully the slides are self-explanatory. Reach out over Slack. If not, we'll try to help out. And then next Tuesday, we'll pick up back with lectures again. All right. Take care.
Thank you.